Okay, so today's topic is operons. We briefly talked about it the day before yesterday in period 6, but I'm pretty sure the other periods you guys already know what an operon is. The first model we're going to learn about is the repressible operon. So an operon is something in prokaryotes only, and they are models of how a bacteria can regulate its genes. So in eukaryotes, humans, what we've already learned, each gene has its own separate promoter. So RNA polymerase will come in and do that gene, see the ta-ta box, do the gene, and then be done with it. And then each gene will have to be then transcribed separately. But in bacteria, as we can see in the model that I've already put the base of on the screen, these 5 genes in this sequence only have one promoter. So this whole line here, this pathway, is controlled by the one promoter, and each of these genes codes for a separate enzyme that makes one molecule in the end. So in that way, the bacteria is more efficient in this process. So the operon itself is this whole area here on the DNA loop. It is the promoter and then these 5 genes in the pathway, but it could be more genes, could be less. It just depends on the pathway. So this here is just a diagram of a segment of the DNA. We're going to want to focus in here for the beginning of the drawing, but make sure you have this as the base sketched out here. The dots here just symbolize that there can be space in between this section here and then the operon here. So let's jump into the operon itself. So the repressible operon, this model, is showing how we create the molecule tryptophan. These genes themselves do not code for tryptophan. They code for 5 different enzymes, and those 5 enzymes then edit the substrate into tryptophan as the end product. So they don't directly code for it, but you can think of it that way just to simplify. But understand that they each code for a separate enzyme. And the end product will be the tryptophan molecule that is created when all of those enzymes edit the substrate. So the segments of our operon here, we have the promoter at the beginning, but something different that we have in this red, orange, whatever color that is the operator. It is a segment of the promoter, and we'll see how it works in a minute. And then we have the 5 genes here. So normally the RNA polymerase, it will come in like usual, come here, and it will go, and it will transcribe the genes, make the RNA, and that'll be that. But let's say, hey, I do not need any more tryptophan, I'm done. How does the bacteria say, I'm done, I need to stop it? And that is what the repressible operon does when we need to stop it. So what we need to understand, we're going to come over here to this whole separate gene over here. It has a promoter. It is called the regulatory gene. So what this gene does when it gets transcribed by the mRNA and then translated by the ribosome, it will create something called a repressor. Let's label that here, repressor. So this regulatory gene creates the repressor, but the repressor, when it is created, is in its inactive form, and we'll see why in a minute. So why did we create this repressor? Well, they are being created just as they are. They're just being created in this inactive form, but eventually they have the goal of shutting off this operon over here. So as the tryptophan is being created, the concentration is going up of the tryptophan. And eventually, the cell does not need all that tryptophan and there's excess. So what happens is that tryptophan, I'm going to change the color. I probably should have done that earlier. Let's make it this purple color. It comes in, tryptophan comes in to the repressor and it binds to it. I just made a copy of it there. as we have learned, um, when a molecule binds to another molecule, it changes its shape. So once the tryptophan binds to the inactive repressor, it is going to form a different shape. So now it is a square, like little head on it. Now, what does this active repressor do? You're probably wondering. So, this active repressor, it comes over here to the operator. Now, we didn't draw it in before, but, let's see here. When we take this is the shape of the operator. It has this square little hole in it. And as you can see, this active form of the repressor fits perfectly into this operator. Now what happens from there is the operator will then change shape. Once again, because we are binding a molecule to it, let's just say it gets a little wider here on the edges. Now when RNA polymerase comes back in, it physically cannot fit over this operator. It is too big. It's a little messy here. It's getting messy, but hopefully you can see the vision. It physically cannot fit over this operator, so therefore the DNA cannot be straightened and unzipped. It cannot be transcribed. It cannot be translated, and that causes the tryptophan molecules to be lowered in production because we're not making them anymore. It literally, the transcription cannot occur, therefore translation, so our production of tryp falls. Now because tryp here is a product in this operon over here, but it also serves to activate our repressor over here, it is called a co-repressor. Let's say the bacteria is like, hey, we need some more tryptophan. How do we remove this? It's actually pretty simple. Since the tryptophan levels over here are falling, the cell naturally needs some more. So what is it going to do? It's going to come over here to this tryptophan molecule right here and break the bond. And because we broke the bond, the shape of this, the repressor is going to go back to that spherical form that we draw before. Therefore, it will not fit. So its bond will break, and the operator will then shrink back to its normal size, and the RNA polymerase can go as usual. And there it goes. So now we broke off our tryptophan because the levels were low, and now the RNA polymerase goes, and the levels go back to normal. Some more things to note. That's it. That is the model and the process of repressible operon. Here's some more notes I wrote that I didn't say. So what we call this segment of the so when we make the mRNA, they aren't separate strands. They're all going to include these genes on it because they are all being translated at the same time. So it will just be one really long mRNA. That was bad. It is called a polycistronic mRNA because it has multiple genes on it. Put that here's polycistronic mRNA. And another thing with repressible operons, the proteins that are being coded here, the molecules, are usually endergonic reactions. We're usually building things in these repressible operon pathways, and they are usually on. They're usually active most of the time in the bacteria. It's almost always on, and it's rarely turned off. So this process doesn't happen often, but when it does, it does happen at some point. Hopefully that made sense. I'm going to keep this basic model. We're going to use it again when we talk about inducible operons.